Hi students, good morning. Today's our topic is carbohydrates and proteins, how to write the structure of them and what are the products we are getting on the hydrolysis of carbohydrate and protein. Okay, and to study what are the monomers. So first we'll look at this. We know that carbohydrates and proteins are natural macromolecules. Last class we studied nylon and terylene. Okay, they are synthetic polymers. That means man-made. Carbohydrates and proteins, they are formed by condensation polymerization. We have two types, one is addition and another one is condensation. So condensation polymerization means the monomers join or react together with the elimination of small molecules like H2O and HCl. So example for natural condensation polymers are carbohydrates and proteins. We know that polyester and nylon, they are also formed by condensation polymers. What about PTFE, PVC, polyethylene? They all are formed by addition polymerization. Now we'll just see carbohydrates. Carbohydrates, the monomers are uh, sugar molecules. Okay, they are actually polysaccharides. That is not there in your syllabus, that's why I didn't mention. Carbohydrates are polysaccharides. So for example, I'll take a example of a carbohydrate, that is starch. Okay, starch is a carbohydrate. It's a polymer made up of hundreds of glucose molecules. Glucose is otherwise known as sugar, okay, which are simple sugar. So monomers of starch. So you have to understand, starch is an example of carbohydrate. So starch is a polymer. It is made up of monomer called glucose. Glucose, you can think it as sugar. Okay. Now, this is the structure of polymer carbohydrate. So how we write that? First we'll write, I'm using two monomers here, okay? See here, these are the monomers. Glucose is represented like this. This rectangular box represents so many CH2 molecules. We don't have to write it, we'll just represent like this. And on both the ends we have OH. This is same like we studied the diols. So what happens? We are going to remove water. I wrote here minus H2O. That means when these two join, you'll remove OH from here and one H from here. Okay. So we remove that and see here in the middle you got O. Now in this side, we have to think that this H is going with some other molecule and here this H is going with some other molecule. You have to imagine in your mind like that. Okay, or if you didn't write here also, it is correct. So this is the structure of starch or carbohydrate. So it is an example of condensation polymerization. So if I ask you to draw the structure, you have to draw this. And what are the monomers? It is glucose or sugar. What type of polymerization? Condensation polymerization. Now we are going to proteins. Proteins are example of polyamide. They have amide link, or usually in proteins we'll say it as peptide link. Peptide means CONH. The CO means it is C double bond O NH. First we'll see the structure, the monomer of proteins. Monomer of protein is amino acid. What is the name? Amino acid. Amino means we have already studied, you have heard diamine, dicarboxylic acid. So amine means NH2 group is there and acid means COOH group is there. You can write the C like this, better you write like this. If you write the opposite also, it's correct. So amino acids, okay, the proteins, the monomers are amino acid and we represent the monomer like this. So from the word we can understand an amino group is there and an acid group is also there. Next, how we are going to write the structure of protein. See, I wrote here COOH in the displayed form and NH2 in the displayed form. So you can understand that this OH and this H is removed as water. So just connect it. You can try this, and you have to imagine this OH is removed and this H is removed. Every time we should have the valency on the open ends, or we call it as valency on both the ends of the polymer. That means this polymer can add more monomers into it. The CONH bond is the amide bond or amide link, or usually in proteins we call it as peptide bonds. So otherwise, how we can represent the structure? Sorry. Okay, so if I ask you to draw the structure of proteins, you have to draw this. So if you check this structure, you can see there is a big similarity, or it's the same structure of nylon. But here you have to think nylon, the monomers were different. It was diamine and dicarboxylic acid. You can take your notebook and check it. The same structure you have for nylon, but here it is a natural polymer and the monomers are amino acids. So how question can come? I can give you this structure and ask you, this is an artificial polymer or a synthetic polymer, mention the monomers of that. If it is a synthetic polymer, it will be nylon, and the monomers are what? Dicarboxylic acid and diamine. If the question is like this, identify the given natural macromolecule, or natural, usually we'll say it as natural macromolecule, and what are the amino acids. So you have to think about the word natural macromolecule. So this will be what? Proteins, and the monomers are amino acids. Now we'll see the hydrolysis of carbohydrates and proteins. Hydrolysis means it's a reaction with water or a base. So starch on hydrolysis. Here I wrote carbohydrates. Starch is an example of carbohydrate. Starch on hydrolysis in presence of a catalyst, amylase, will give you the monomer of starch back, that is glucose. We know that starch is made up of glucose monomers. So when you do the hydrolysis, we don't have to write the equation or anything. So you have to just know that hydrolysis gives you back the monomers. So proteins, if you do the hydrolysis, we will get what? The monomer of protein, that is amino acids. So you have to just know about the catalyst which we are using. This is enzymes, okay? Amylase and protease. You will be familiar with this in bio. So I hope that this all is clear. These all are clear for you. Then we have the questions. Please write all the questions in your notebook and put today's date, and you have to complete it. Thanks.